In section D, we're going to look at the other method of reproduction, of cell division that occurs within our cells, known as meiosis. So we're going to define terms, the process of meiosis. We're going to look at things like homologous pairs and alleles, karyotypes. We're going to differentiate between haploid and diploid, and then explain the significance of some of the things that happen during meiosis. So why are we looking at meiosis? Well, again, remember, this whole chapter has been centered around a couple specific questions. Remember, Angelina Jolie carries the gene that increased her risk of cancer, so could she pass that risk on to her children? Are Lance Armstrong's children at an increased risk of cancer because he underwent cell-damaging chemotherapy prior to their birth? In order to answer those questions, you need a better understanding of the type of cell division that produces sperm cells and egg cells that are then used to form a new, or used to form offspring, the zygote, which eventually becomes the new baby. So this is known as meiosis. So meiosis is a specialized form of cell division, and this cell division occurs in the gonads of an organism to produce gametes. So when we use the term gonads, male gonads are the testes, and they produce male gametes known as sperm cells. Female gonads, these are the ovaries, and they produce female gametes that are known as egg cells. Now, one very important thing to note about meiosis is that in order to produce these gametes, what meiosis is going to do is it's going to reduce the number of chromosomes in each cell by 1/2. So if you take a somatic cell, remember somatic cell is just a normal body cell, it contains 46 chromosomes. Okay, so that's the normal number of chromosomes for a normal body cell or somatic cell. What meiosis does is it produces gametes that only contain half of that number, which would be 23 chromosomes. Now, one really important thing to note about somatic cells is that in order to make those 46 chromosomes, chromosomes are actually found in pairs. These are called homologous pairs. So a lot of times you'll hear that the normal number of chromosomes for a human cell is either 46, or sometimes you'll hear the term 23 pairs, because the 23 chromosomes, they're paired up, which gives you that 46. Now, a couple things to know about homologous pairs. That prefix homo means equal or same. So homologous pairs are chromosomes that are the same size, they're the same shape, they carry out the same genes. So they're carrying out the same sequences to build the same proteins. The only thing that's different about these homologous pairs are the versions of these genes that they may carry. This is the term known as alleles. So for example, if the gene they're carrying is hair color, one might carry the allele for blue eye, for hair color. One might carry for blonde hair, the other might carry for brown hair. Or if it's carrying the gene for eye color, one might carry the allele for blue eyes, another might carry the allele for brown eyes. And so when your cells are forming gametes, each gamete only gets one of each pair. So if you had 23 pairs, or 46 chromosomes, if the gamete only gets one, then it gets half as many, which gives you that number of 23. Now, in genetics, we use these things called karyotypes. Now, karyotypes are just photographs of chromosomes that are arranged in pairs. And so we just said a normal cell is going to have 23 pairs of chromosomes. 22 of them are what we call autosomes. And these are what determine every trait about you except for the very last pair, which are known as sex chromosomes, and they determine the sex of the individual. Now, in lab, we're going to look at karyotypes in a little more detail. We're actually going to make karyotypes and determine if it's a normal or a genetically mutated individual. Now again, gametes, it's the collective term for sperm cells and egg cells. Specialized sex cells, we said they're produced by meiosis in the gonads, so either in the testes or the ovaries, and they produce haploid cells. Haploid is the term that we use to describe only having one set, or 23 chromosomes. So when we refer to egg cells and sperm cells, we also refer to the fact that they're haploid, meaning they only have half as many. So they only have 23 chromosomes. Why is it important for sperm cells and egg cells to only have half the number? Well, keep in mind, the purpose of sperm and egg cells is to eventually fuse to form a zygote. One carrying 23 chromosomes, the other carrying 23 chromosomes, and when you combine those, you get that magic number of 46, which is the normal number for a human cell. So we use the term haploid to describe having half as many. We use the term diploid to describe having both sets of chromosomes. So we say that a zygote is diploid because it contains two sets of chromosomes, one that came from the mother, the other that came from the father. So let's look at how these gametes are actually produced. It's a process called meiosis. And so what happens here is the cell goes through normal interphase. So it still goes through G1, still goes through S, still goes through G2, copying that DNA, copying those organelles, growing and getting ready to divide. Then the cell moves into the two stages of meiosis. Now, you'll notice on the screen that there is a thing called meiosis 1 and then something called meiosis 2. So the 1 is for the first division. The second is for the second division. So unlike mitosis, where the cell only divides once, meiosis is going to divide the cell twice. In meiosis 1, the whole purpose is going to be to separate out the homologous pairs. We're going to talk about this in a little more detail in just a moment. And then meiosis 2 is going to separate the sister chromatids into 4 haploid cells. So general process here is to take a cell, to divide it twice to give you 4 cells that are all haploid that can become gametes. Now this is the overall process of meiosis. If you look on page 122 and 123 in your text, you'll see meiosis 1 and then you'll see meiosis 2, and so we're going to break these into their specific phases. Now what's good here is that you'll notice that the phases of meiosis are the same as the phases of mitosis by name. So you still have PMAT. The only difference is there will either be a 1 or a 2 following each phase. So prophase 1 begins meiosis 1 by doing some of the same things that we're used to seeing in prophase. So the nuclear envelope breaks down, nucleolus disappears, microtubules assemble, or those filaments. We see DNA condense, so we can see chromosomes and they're able to move without getting entangled. But one big thing that characterizes prophase 1 is not only do we see chromosomes, but we see homologous pairs of chromosomes. So the chromosomes are going to find their homologous pair, they're going to pair up, and then each pair of homologous chromosomes is going to undergo something known as crossing over. So what crossing over is it's actually where these homologous chromosomes pair up and the chromatids literally crisscross over each other. By doing that, they allow these chromosomes to exchange genetic material that will increase the chances of a variety of gametes. So crossing over helps to increase genetic diversity because once these homologous chromosomes cross over each other, when they're pulled apart later on, they're going to be genetically different than they were before they crossed. So prophase 1, again, you see the nucleus disappearing, you see chromosomes become visible, but not just any chromosomes, we see homologous chromosomes pairing up and crossing over. Then when they move into metaphase 1, again, we typically associate metaphase as moving to the middle. Same thing here. But again, the big difference here is that in metaphase 1, it's not just chromosomes lining up in the middle, it's the homologous chromosomes that paired up. Remember, they had crossed over. Well, now the pair moves to the middle. And they experience something known as random alignment. Random alignment basically says that when these homologous pairs go to the middle, they can line up in any kind of random arrangement that they want. And so again, because of random alignment, just like crossing over, it increases the chances of producing genetically different gametes, which increases genetic variety. And so you look on the screen, you'll see that this is one possible way that these pairs could line up in metaphase 1. But look at the bottom image, this is a different way they could line up. And so you'll notice that if we were to take this first cell and split it down the middle, you end up with 2 cells, and that would then split into 4 cells. And so the gametes would look like this. You see their chromatids in the center. But because of random alignment, that they can line up in a different way, this cell, when it splits twice, would produce these 4 different gametes, which are obviously different than these 4. So random alignment increases genetic diversity. Now, once the pairs have lined up in the middle, the cell then goes through anaphase 1. Again, anaphase, typically we have a splitting, a separating, and a moving. So again, same thing here, except it's the homologous pairs that separate and move towards opposite poles. And then telophase 1, the new nucleus reforms around those chromosomes. So at the end of telophase 1, we have one cell that has two nuclei, and then cytokinesis occurs to split the cell in half. So at the end of meiosis 1, we have two cells that are genetically different from each other, and you can see here. So this is prophase 1, homologous pairs pair up and they cross over. Metaphase 1, they randomly align the pairs in the middle. Anaphase 1, where the pairs split. And then telophase 1 and cytokinesis 1 that splits into two cells. But again, we're not finished because the cell then goes through meiosis 2. Now one key thing to note here is that meiosis 2 is virtually identical to mitosis, because where meiosis 1 focused on all the events around the homologous pair, meiosis 2 focuses the events around sister chromatids, which is what mitosis did. So in prophase 2, we see the nuclear envelope breaks down, the chromosomes become visible, microtubules lengthen and attach to those chromosomes. Metaphase 2, the sister chromatids are going to line up in the middle. In anaphase 2, the sister chromatids are going to split, move to opposite poles. Telophase 2, the nucleus is going to reform around these. And at the end of cytokinesis 2, the cytoplasm is going to split those cells into 4 daughter cells that have half the number of chromosomes, so they are haploid, and they are all genetically different. So you see here is prophase 2, metaphase 2, anaphase 2, and then telophase and cytokinesis 2 splits into 4 cells, and when you look at those chromatids on the inside, you'll notice that they're half what they started out with, so that's haploid, and that they're all genetically different than the rest. So here gives you a good comparison of mitosis versus meiosis. Mitosis producing 2 daughter cells that are genetically identical, meiosis producing 4 daughter cells that are haploid and are all genetically different. So the question is, can people pass cancer on to their children? Well, in Angelina Jolie's case, we know that genetic testing showed Angelina Jolie inherited the gene from her mother, and so because it was something that she inherited from her mother, it's something that she could pass on to the egg cells that she produces. But remember, egg cells only carry half the number of chromosomes. So because of that, each of her biological children have a 50% or a half chance of carrying the mutant version of this gene. Now in Lance Armstrong's case, the cause of his cancer was not clear-cut. And his cancer, it could have developed in response to inherited mutations, or it could be exposure to carcinogens that cause cancer. There's also the concern that it could increase cancer risk because of the fact that they use chemotherapy. So as far as the risk of him passing on testicular cancer to his children, it's not possible to determine the risk there.